Hello, my name's Callum and I'm representing the Dundee University of Life Sciences, and I'm here with Professor Labib of the Medical Research Council Unit. It's a pleasure to meet you, sir. Thank you for coming to us.
That's right. So, hi. I've been here in Life Sciences in Dundee for about a year now. I came up from the University of Manchester before. And my group's interested in one of the most fundamental issues in biology that really goes to the heart of our understanding of what living creatures are, how they're able to reproduce themselves, and how our bodies are able to protect us against genetic diseases such as cancer.
DNA is essential for cell function and efficiency.
So cells have to make one perfect copy of the chromosomes before they cut themselves into.
And just like a zip has a fastener to zoom along the zip opening and closing, the cell needs to have a molecular machine to unwind the two strands of the DNA so that each can then be used as a template to build another.
So the key to this process is that the cell must only unwind the DNA once, because every time it unwinds it, it could make another copy, and cells must only make one copy of their chromosomes in each life cycle of the cell.
DNA replication is carried out by replisomes.
The ribosome's the larger machine that does the whole business of copying the chromosome. So it's actually called a helicase, and it's that molecular machine that we study.
Replisomes are assembled by CMG helicases.
So most of the work in this area in our field in the last 15 years is how the cell puts the fastener on the zip, which allows it to unwind the DNA. So we know quite a lot now from the last 15 years about assembling the helicase that unwinds the DNA. At a certain moment, the cell has to build the fastener, the helicase, on the DNA, so it gets one unique chance to unwind the DNA, and when it's completely finished, it has to be taken off so that it can't happen again.
Replisomes are disassembled by CDC48 and ubiquitin ligase.
So we needed a stroke of luck to understand how the helicase is taken off the DNA at the end of the process of copying the chromosome. And it turns out that the cell puts a little tag on the helicase. It's a little marker that tells the cell that this is something that's going to be destroyed afterwards. It's actually another little protein called ubiquitin, which is stuck onto the helicase. And when it's on the helicase, another protein, another little molecular machine comes along and pulls the machine to pieces, and that's how it's disassembled from the chromosome.
So the cell has to make sure that it only disassembles the helicase that unwinds the DNA at the very end of the process. So for the future, we want to understand how does this disassembly process really work in detail, and we'd like to recreate the process in the test tube ultimately so that we really know that we understand how it works. And then we want to understand how do cells make sure that this never happens until the very end of chromosome replication. Otherwise it would be very bad for the cell because it would be trying to divide itself before it's actually finished copying its chromosomes, and then it wouldn't be able to give its daughter cells all the genetic information that they need.
Mutations in replisomes can result in diseases such as cancer.
So chromosome replication is one of the processes of basic biology that goes wrong very early in the development of cancer. So in almost all human cancers, one of the first things that goes wrong is precisely a defect in this process of copying the chromosomes.
This research could be useful to develop medical treatment for various diseases such as cancer.
So many mature cancers in our bodies retain these defects, these inherent defects in chromosome replication. And that's potentially very useful because it represents a kind of Achilles heel that we can exploit to try and find a drug that would hurt the process of chromosome replication a bit, so that the normal cells in our body can cope with that small defect in chromosome replication, but the cancer cells, which are already defective in the same process, can't cope and they die.